Hi guys, welcome back to the channel. So in the last video we have seen how to design the TETRA-ARMS PCR primer using the different softwares, right? So today we'll be seeing how to check whether they are specific or not, otherwise we'll be getting too many bands when we do the PCR and the gel electrophoresis, right? So if you remember, this is the RSID or the variant that we have used in the last video, so we'll be using the same RSID again to check the specificity, right? So from that output, if you remember, if you have practiced, you must be knowing that we had got some 10 outputs, right? So from those 10 I have selected one to further process my primers, right? So I'll just go there and so this is my Excel sheet, and I have pasted the primers from output number 1 here, and as you can see I have made different columns to and name them properly so that it's easy for me to understand, right? Product size, TM is melting temperature, that I'll be writing afterwards. This is the product size that I'll be mentioning, and here is number of bases, right? Don't get confused with this 27 because it is showing as there are extra spaces. I have written 5 dash and 3 dash just to show you so that there is no confusion, right? So here there will be three products that will be produced, and that are used in your PCR, conventional PCR for TETRA-ARMS. So the product will be IF and OR, okay, the combination of these two. Next one will be of OF and OR, and next one will be of OF and IR, okay. So without wasting any more time, we'll start how to design it. So this is the software that we'll be using, that is called as MFE primer 3.1, right? So you just need to paste your uh, sequences here and check the product size and the specificity, right? So first we'll start with the OF and OR. So I'll come to my Excel sheet, control copy, paste it here, followed by OR. Again copy from here, paste it just below the OF, right? So I have pasted it here. So now I don't need to change anything extra, just I'll go down and click on run. Okay, so it is showing, it is running the specificity test and it will show you the results. So here you can see P1 is our OF and what we have fed, and P2 is the OR. It is showing me the base pair size, so base is 23 and here it is 20, so you can also count it, right? Minimum bases that are generally used is 18 in a primer, maximum goes to 25, more than that it is not suitable. And GC content is showing 52 and here it is 60, so this is also fine. Generally GC content is from 45 to 60% is considered good, and some cases 65 is also fine. If it is around 50 it is okay, so here also it is not an issue, it is fine. Now here I'll see the temperature, that is the melting temperature. It is showing 63 and it is showing 62 for OF and OR respectively. So this is also fine, we can proceed with this, I think. And melting temperature is usually you will find from 55 to 70 degrees, so 62 is usually the ideal one, so this is also fine. And one more thing, make sure both the primers what you are designing, the difference between the TM of these two must be within 5 degrees, right? Plus minus 5. So if it is 63, it should be, the other one should be max to max 68 or below, that means it will be 58, right? So accordingly you can calculate if I am wrong also. Now we'll see the potential amplicons. Usually we expect there will be only one amplicon, and we already know that our gene is on chromosome number 10, and we are expecting one potential amplicon, but it is showing us two, right? So but if you look here carefully, it is showing on chromosome 1, I mean one first number is chromosome number 10, and it is showing product size of 500 base pair, and the melting temperature is 64 and 63, right? So basically forward primer 64, reverse primer 63. But for this one, it is for chromosome number 11, it is showing base pair size 872, and the temperature, I mean the melting temperature, is also 30 or 33. So that means we can use this primer, as it will not be hindering our process, as the temperature that we'll be using in the primer PCR polymerase chain reaction will be 64 most probably, and we have to set up a gradient also, temperature gradient, to check where it is working properly. So we don't need to change anything here basically, so we can go with this, and we'll note it down from here what is the size, that is 500 bases. So P1 and P2 means OF and OR, right? And this product will not be formed until unless this temperature, that is 30 or 33, is provided. Since our temperature that we'll be providing is 64 to 63, in between these, nearby plus minus 2 degrees, so our product size that we will see in the gel is also be going to be nearby 500, and usually it will be 500 only, right? So I'll just take this information and note it down in my Excel sheet. So I have got the result, product size for OFOR is 500 base pair, right? So I have written it here. Same way I'll check for OFOR also. Okay, this is fine. Temperature I have to write down. So for OF it is showing me 63. So which one is OF? This one is OF, right? So I'll write down 63.28, right? And for OR it is showing me 62.67. 62.67. This is useful to note down, as when you are referring it in future, it will be easier. Otherwise you have to again go to the software. Sometimes the software is too much in use and it gets hung up, and you have to wait for a long time. So better to note down all the important information, right? So this says it is fine. Now we'll go for the next one, that is IF and OR, right? So IF is your, as you have written here also, normal allele, right? So how do I check the specificity, whether this is making a specific what do you say, product or not? So what I have to do is simply copy IF from here. So this is 5 dash and this is 3 dash n, right? So I'll just click here and I'll start copying. I have showed you in the last video also. Shift, then press the arrow key, copy this, paste it in the software. Next one is OR. So Control C, paste it just below the IF, right? Now here there is one new condition. What happens here is, when we are checking for the specificity IF and OR, see, whenever the software designs primers for you, it will design it uh, with the mismatch, right? So mismatch means it will induce or add a mismatch at the 3 dash position, okay, in this side, on the in IF of course, and not in OR, in inner primers only, right? So in this condition, it is already at what I have taken here, it is already having a mismatch, right? So how do you check it whether it is having a mismatch or not? Very easy. You open your gene sequence what you have downloaded, and it is usually the raw gene sequence and without any mismatches, okay? So we just focus on these three selected parts, 3 dash n, and here 3 dash n is from here, right? So you can see the difference is there, right? Here it is saying CCC and here it is saying CCA, right? And since it is in forward direction only, no need to reverse complement in this case. So you can see the mismatch is inserted at the 1 2 3 third position from the 3 dash end. So mismatch is, you can see here in the primer it is showing you C, but here in the original genome sequence it is showing A. Okay, that means the mismatch has been inserted here. So why a mismatch is added, I'll give you a general idea. You can read more about it to make it more specific, right? That is why we add mismatches, and more you can read on Google and maybe in the notebooks if you read it, right? So this is how the mismatches are added. So what I have to do now, whenever I am going to run it with the mismatch, that is the unaltered, okay, inner forward, I should be getting 0 amplicons, right? In this case, why? Because you will see it here. So I'll just keep it as such and I'll run. So it has run and it is showing me 8 potential amplicons, but I just now told you that I should be getting 0 amplicons, right? So here what it shows is my gene, I mean this RSID is on chromosome number 10, right? And the IF, that is this P1, is showing me melting temperature at 63, okay, and P2 is showing me the melting temperature at 62. So that means if I want to check whether the uh, primers, I mean these potential amplicons are correct or not, what and how I will be checking is I'll check what is the TM as well, right? So if the TM is near to my to this TM around 63 or 62, or plus minus 5 degrees, then I'll see if it is a potential amplicon. So we'll check here. TM you can see 40, 35, 34, 31, 31, 39, 35, 30, right? So out of 8, all of these 8, none of them is above 40, right? So that means when I run the reaction at 60 or 61, I mean 62 or 63 temperature, there should be no issues, as my primers will be getting aligned only at 62 or 63, right? So when I give this particular specific temperature, this bond will be formed between these two. I mean, of course not here because a mismatch is there, right? So when the mismatch is removed, then only it will be uh, making the primer, right? So this one is perfectly alright for me to proceed, and you can check the GC also after checking the TM. GC is showing 61 and here it is 60. I told you it should be from 45 to 60 or 65, it is fine. So this is fine. Now in this case it is fine, but if I need to reduce the GC, okay, so before that I'll just note down the data, or maybe when I uh, add the remove the mismatch, then I'll add it, right? So basically what the literature says is with mismatch, that means in this condition it should be showing me 0 amplicons, right? And as you can see on this particular temperature there are no amplicons, right? That means what I have designed till now is correct through the software, right? So now I'll just uh, go back and again what I'll do is I'll again copy IF here and next one is OF. This one is that, right? So now I need to add the mismatch at 3 dash and here. Okay, I have to remove the mismatch. So when I remove the mismatch, this should form a product, right? So after removing the mismatch, the product should be 1, right? As you know, in the ideal condition it should be 1. So we'll see what mismatch is there, what we have to add here. So if you see here, you can see my cursor. I'll be highlighting it here. You can see this red colored C, right? So from here, 1 2 3 C C C 3 dash n, and from this side this is 3 dash n C C A. So this is the mismatch basically, okay, that is inserted here in this primer. Okay, this is with mismatch, and this is the normal sequence. So I'll just replace this C with A, right? So I'll just click here, delete this and write A, and I'll give it a run. It should show me 1 amplicon, right? You will see. So it is showing me 4 potential amplicons, right? So is it correct or not? We'll see. First we'll see the temperature. Melting temperature is 60. Here it is 62. It is fine. GC content is also fine. Base pair size is also fine. No hairpin list, no dimers. Now 4 potential amplicons. So first thing to remember is our chromosome is on, our product is on chromosome number 10. Fine. Other than that, RTM is approximately 60, 62. Okay, nearby 60. Here, out of these 4, only one shows me the temperature around 60, 62, 63, right? Other than that, these 3 are below that at 35, 35, 34, 30, 33. So that means these, when I run the reaction at 62 or 63 degree temperature, after setting the temperature gradient, it will not be binding to any other uh, basis. That means I'll get a proper product of this 211 base pair. That means the primer that I have designed is correct. Only thing I need to be careful is about I should not be running at lower temperatures, otherwise there are chances of these non-specific binding, but this doesn't happen, and I'll show you in later videos how to avoid all these issues. But today we'll just focus on designing the primers and checking the specificity, right? So here this is the correct answer that I have got. So I'll just note it down in my Excel sheet. So product size of IF and OR, okay, equals to 211 base pairs, right? Fine. So this is also correct. I'll write down the if you go below here in the software, you can see what all is and how the matching is done. GC content, the total is 48. That is also fine. Okay, so I'll just note down the size and all. Size is shows 21. Okay, and here it is showing 20. Fine. So I'll write down the TM of IF. So first one is IF, that is how much it is, 60.66, right? And for OR, okay, same, right? So it doesn't change, of course. Now in this case we have GC content as 57 and 60. Suppose I need to reduce it in any case. If it is there, suppose it is around 75% or 80%, so what do? I will be deleting these what do you say, nucleotides from the 5' end, that is from the left hand side. Okay, in any of the sequence, if I need to delete, not from this side, always from the 5' end. Okay, I'll just delete one. Okay, and I'll try to run it. For example, I'll show you here. I'll just delete 1G, okay, from the OR, and I'll run. So it will be showing you the changes. right? As you can see now it has become 57. The GC has become exactly 57, 57, right? Potential amplicons is of course the same because there is a minor change, but the GC has reduced. Okay, and as well as TM also reduced. Initially it was 62, now it has 59, right? So GC basically it has 3 bonds. So if you reduce GC content, your TM will also be decreased, and if you increase the GC content, TM, the melting temperature, will also be increased, right? Same thing. So now that is for IF and OR. Now I will check specificity for OF and IR. So this is IR means our mutant allele, right? So that is also same, but a little bit difference is there. So we'll see how. So till now this is clear. Oh, I hope. So Control C, same way I paste it here, and below that I want to add IR. So this is my IR. Okay, Control C. So now there is one thing to be noted here is that whenever you are checking your IR, this is reverse, right? So first you need to make it forward so that you can read it properly. So what we use in the last video, we used reverse complementation, right? So we'll go to this reverse complement, paste it here, submit. So this is the reverse complement, right? Control A, control. Fine, this is here. Now I need to check uh, with and without, right? So first uh, I'll check it without changing it, right? I made the reverse complement also. I'll just paste it as such, and I'll click on run. Right, so it is showing me how many 13 amplicons, right? Now you again note down the temperature, TM 63, 62, right? And here you see the temperatures 1, 2, 3, 4, 5, all are around 30, 32 like that, right? So that means there is no issue, we can proceed, right? Initially when you are learning, this is more than enough. Okay, when you become little bit expert, that you can then you can explore many things, but this is more than enough for now, right? Now I'll check. This is with mismatch, right? I just took it from the Excel sheet directly and pasted and checked it with mismatch. Okay, ideally it should be showing 0 amplicons. Okay, and since none of these the results we saw was going to be in our range, so that means I can consider it as a 0 amplicon, right? Now I'll check after removing the mismatch, what does it show. So now here's the trick, little bit tricky part. So what I'll do is I'll just click here, control, find it from the 3 dash and to check whether this is the correct sequence or not. So I'll just delete 1, 2 and 3. So after I deleted the third nucleotide, you can see this C 1, 2 and 3 till T I have deleted, and this is matching. That means these three are having some changes, that is going to make it more specific, right? So basically if you see here, I'll write it again. You can see here the first letter here is T, right? But here it is C. That means there is one mismatch. Here second is G. Here it is also G, right? So this is fine. And then there is one more mismatch as the third position, right? Here it is G, and here it is T, that is highlighted in the blue color. And then again the same repeated parts. So that means we have inserted two mismatches, that is at first position and the third position from the 3 dash end. So here what we need to do is we just need to make the complement of these and add it to the software that we are checking for specificity, right? So how we'll do it, and this is how we'll be doing it. So this is our IR, right, and this is the 3 dash end. Fine. So in place of C, what we will be adding, the reverse complement of this, okay, and after removing the mismatch, so it will become G, and in this position it will become A. Fine. So you can see here very clearly this T is the mismatch and this C is also. So when we come here, we have added the mismatches at 3 dash and first position. I mean, we have removed the mismatch, and same way here also. Just need to now run it, and so it is showing me two potential amplicons, right? So now you see at the look at the TM 63 and 61, right? Now you go down and check for the potential amplicons. Both the products at chromosome number 10, size is 329 and 327, temperature is 64, 62, 59, 62. Okay, so this is fine, right? So even if I get both of these are very nearby. Okay, so even if both of them are present, you won't be able to recognize them in the gel as it is very much nearby. So even if I get this product, or even if I get this product, both are okay for me, because the temperature range is almost similar, right? And both are on the same chromosome, product of P1 and P2. Fine. And GC content is also in the range 52 and 60. So that means what we have made is perfectly alright, and we can go with this. So I'll just note it down in my Excel sheet. One second. So here it is. Size is, it is showing me two sizes, 329, 327. So I'll write down both, 329 base pair and 327 base, right? Fine. And the what is remaining size, I mean the TM of IR. So this was my IR. TM is 61.8. 61.8, right? And these things you can use a formula to set up, or you can write it manually also, right? So basically now this way I have checked the specificity and have written the base pair size also. So once this is done, you can use this data to send it to the, you will send it to the primer designing company, to the vendor. They will design it for you, and when you run your PCR, you will get to see the band, different kind of band. So if the it's your blood sample or whatever samples you're working on, if it is having that mutation, it will be forming this band, of course. And in general, OF and OR will present in each and every human being, of course. And this one will be present, this one will also be present, right? 211 band as it is normally, right? So this way you check for the specificity, and you can apply it for your studies. And I hope you understood it, and if you have any doubts, you are welcome in the comments, and don't forget to like, share and subscribe. And more, I'll come up with some more tools to check the same thing, the specificity results of the design primers with different tools. So just stay tuned, and this is just one software. There are many softwares, so we'll have a look on them as well, right? So like, share and subscribe. Bye bye.